butanol ethanol fermentation process, and this was quite important because during the World War I there was a need for particularly acetone to be used in explosives. And so he patented this process here, and he used a bacterium, Clostridium acidobotulicum, which is actually also used today, and many of us actually also have it in our gut. And that resulted in establishment of processes both in the US and in the UK. He was pretty generous and gave the Brits this process here, but there was a caveat to this because he convinced Balfour, which was the Prime Minister in UK at that time, to make a declaration in 1917 in return of this, and that was to establish an independent state of Israel. So here you can see biotech and grand politics already went hand in hand at that time. Another real key landmark in the use of microorganisms for production of stuff was really the discovery of penicillin. It was discovered in 1929 by Alexander Fleming, and here's his famous Petri dish, which was really serendipity that he had left the Petri dish open, so he had a contaminant. He was studying a bacteria you can see growing on the plate here, and when he came back after the weekend or holiday, he observed that there was a fungus growing here, and you can see there was not really bacteria growing around the fungus, and that led him to think that this fungus was probably producing a chemical that kills the bacteria around here. And that later on led to actually commercializing penicillin production, which is probably the most life-saving drug that we have ever seen. And it's worthwhile to think about. And it's used, of course, widely for treatment of many infectious diseases. This led later on to what is often referred to as the golden age of antibiotic. Selman Waxman was a real important pioneer in this. He received also the Nobel Prize in Medicine and Physiology in '52 for discovery of streptomycin. This was an antibiotic produced not by a fungus but a bacteria that were then found actually produce a whole wide range of different chemicals that could be used as antibiotics. And in the 50s and 50s to the 80s, all major pharmaceutical companies were producing antibiotics by microbial fermentation and making a lot of money on this. But as I'll show to you later on, there was really a development. And today, almost all antibiotics is being produced in China and India, and none of the big pharma is in the antibiotic business. It's interesting to see that if we look at the current use of antibiotics, and here's the intake, antibiotics used per capita, and here's the GDP per capita out here. And you can see, except for a few outliers here, that's France, UK, and US, and that's special why there are outliers here, but you can see generally the intake is almost the same, independent of GDP. That is because antibiotics is so cost effective. So even if you're a relatively poor country, you reach the same antibiotic usage as in a completely industrialized country. And that is, again, this, as I said, that we often forget. We focus on many of the new therapies and so on, but antibiotics today are still really saving millions of lives around the world. However, we do have a problem about antibiotic resistance, and I'm not going to talk about that, but there are really problems with actually developing new antibiotics, and very few people are working on this for various reasons that we can maybe discuss later. So this was an example of using microorganisms, but as we heard about also early on in this meeting, when the ability came to suddenly use genetic engineering, that revolutionized the world. And particularly the biotech industry arose because of this, because suddenly you could begin to produce molecules that we naturally produce. And for example, human insulin at that time was extracted from pigs and purified and used for treatment of diabetics. That could now be produced in yeast and also in bacteria. And today we have a whole range of recombinant proteins with market value exceeding something like $200 billion US. And they come in many different categories. And here you can see also that many of them have rapid growth in their markets here. So the concept of cell factories, as we often refer to it, is really widely used. It was traditionally in bread, beer, and wine. It went on to yogurt and cheese and other fermented food products. But today, pharmaceuticals are produced, fuels and chemicals. You can have citric acid every time you drink a Coke. That citric acid in the Coke is derived from a fungal fermentation. 1,3-propandiol, what is used as a chemical feedstock to produce fabrics that, for example, goes into carpets or other types of products or other types of fabrics. And if we look at the total chemical production, it's a huge industry. It's much, much bigger than pharma industry. But today, renewable chemicals, as we refer to it, produced by cell factories, is only about 1% of this, but it's growing much, much faster, as Francis was also telling us about early on, so we eventually can replace petroleum, use of petroleum for this, with a biotech route. I already mentioned the pharmaceutical, biopharmaceutical, which is a very large market and is also growing very rapidly. So how is this done? Well, how do we engineer microbes to do this job? Well, Francis was talking very much about specific enzymes that you can tailor to make specific chemistries. But when we want to make a new chemical, we often had to stitch together a whole biosynthetic pathway involving many different enzymes. And we do this, of course, through genome engineering. We heard yesterday about fantastic tools to do this. So we can go in and modulate and really change the genetic landscape of that cell factory that we want to use. For example, if we want to take yeast here, that is very good in converting glucose to ethanol, we're going to recruit all the metabolic reaction that yeast has and then produce a new product. And it can also be that we take genetic materials from plants or bacteria and we stitch together a whole new pathway here leading to these products over here. And a key challenge is really not only to produce this product, but also optimize it so we can produce it cost competitively. So this idea is often referred to as the biorefinery concept, in a complete analogy with the refining of oil, where we take, in this case here, biomass and degrade it into sugars, and then we have the sugars converted in a microbial fermentation and not just produce ethanol, but produce a whole range of different chemicals that we can find applications for in materials, or it can be in fuels, it can be in pharmaceuticals, food ingredients, and you name it. So this sounds easy. We just put in some genes into yeast and we produce it. But let's look a little bit on the history again here and some of the challenges associated with it. So let's look at penicillin process. So here is how much the original strain of Fleming that he isolated was producing of penicillin here. It was producing very small amount. And actually, when they started the penicillin production process and went into the clinic, they had difficulties in getting enough stuff of this. So they actually took the urine of those patients that they were treating and extracted penicillin out of that and reused it. Okay? We don't do that anymore because they managed, as you can see, to rapidly improve the performance of this cell factory. Then there was a kink here, but still, notice this is an exponential axis here, a logarithmic axis here. So you still have really rapid growth, but also notice the number of years here. It took many years. And so even over those years, you got a very efficient strain, and today penicillin is produced in the range of 60,000 tons per year, and it costs only $8 per kilogram. Very, very cheap product. So, but why did it take so many years, and why is it so complicated to improve this production? Well, this is the answer. This is metabolism. This is what is going on inside these cell factories. And for example, if we take yeast cell, it has 2,000 metabolic reactions, and they're associated with 900 genes. So it's not just a single little thing that we need to change. We need to understand this whole landscape of metabolism and how it interacts. And that's why we like math, because we have to apply math to study these here. There are similar numbers for a human cell. It's more complex, 8,000 metabolic reactions associated with about 3,000 genes. So not only that, you probably heard about metabolism in school and heard about specific reactions. You may have heard about glycolysis. You may have heard about the tricarboxylic acid or the Krebs cycle, that's often referred to. And we hear about metabolism in this organization into pathways. But this is not how the cell works. The cell has not read the biochemistry textbooks. So this is how metabolism works, because many of these individual reactions are highly connected with each other. And what we have here is when we have collected information about these 900 genes catalyzing these 2,000 reactions in yeast, and then we map here the green ones are the enzymes, and they are linked to different metabolites. And here are metabolites that are in the cytoplasm, and here are metabolites that are in the mitochondria, and how these enzymes are interacting with all these different metabolites. And you can see it's extremely connected. And that's because many of these enzymes use common substrates, common reactants in these reactions. And some of these are, for example, ATP and NADH and NADPH. So these are the energy and electron carriers in the cell. But also, if you take other chemicals in the cell, are used in really a large number of reactions. You can take pyruvate, acetyl-CoA. They're used in 50 type reactions across the metabolism. So how can we study such a complex system? Well, lucky-wise, through the genomics revolution, we actually now have a blueprint about what is available in the cell, and this has really changed it. And of course, also when we have now all these data here, we can begin to integrate these, and this is what we call systems biology. So really, with this multi-omics analysis that has made it possible is that we can open this black box here. Well, here it's actually white, but you have a substrate coming into this cell factory. We want to produce these different products out here. But now what we can do is that we can sequence a genome. We can measure all the messenger RNAs, all the transcripts. We can measure all the proteins. We can measure maybe not all the metabolites, but we are getting better at it, so we can measure really a large fraction of these. We can even quantify what is the activity of these different pathways through measuring what we call the fluxes. So all these information here begins to provide us insight into how this cell factory is functioning. It's still complex, but we are having a tool to kind of begin to see what is going on inside this cell. And this is, of course, exactly the same concept we can apply to a tumor cell when it's different. How is a tumor cell differentiating itself from a healthy cell? So conceptually, it's exactly the same that when we study cell factories to when we study human disease in terms of metabolism. And this is what we refer to as systems biology, as I mentioned. So when we talk about developing these cell factories, we go through this design, build, test cycle that Dan Gibson was also talking about yesterday. So we have our cell factory. It can be yeast we're starting with. We do a design. We then build this, and we build this by genome editing. We introduce the different modifications we want to do. We test it. And what is very important is that often this design doesn't work as we wanted it to do. And that's because of this high connectivity in metabolism and complexity and so on. So we're very keen to actually capture knowledge. So I often say that there is no difference between basic research and applied research. It's, of course, very applied. We want to take a yeast cell to produce a chemical more efficiently. But we acquire a lot of knowledge about metabolism and how all these different reactions interact with each other in this process here. So here is kind of how this goes around. We take genes from plants or other sources, and we build what we call a proof of principle strain. We show that this pathway can actually be stitched together, and yeast can produce a few milligrams of this product that we're interested in. That's a good start, but there's a long way to have what I call a final strain, something that can go in and be used for commercial production, because that has to meet some requirement in terms of econometrics. So it has to, for example, have a very efficient conversion of the glucose, because that's a major cost. It has to do it at a reasonably rate, otherwise it will take too long time to produce the product. And it also has to have a reasonable titer, because otherwise it gets too expensive to purify the product. And that takes a long time. I told you about for penicillin. So today we can do it faster, but it's still, a multi-year effort with many, many people involved. And typical cost is about 50 to 100 million US dollars just to develop the catalyst. So we are working on developing novel technologies that can speed this down, bring down these costs, so we can develop these catalysts of the future much more efficiently. So we focus to work on yeast. Who won't want to work on yeast? I mean, it's make, as I said, bread, beer, and wine, and what a better starting point can you have? But there are other reasons. I'm going to get to those. So we take yeast, and we are then engineering yeast to produce a whole range of chemicals over here that can find different applications. They come in different categories and therefore have different challenges associated with this. But you can say that the challenge here is to demonstrate if can we take one organism and can we reprogram its metabolism to now efficiently produce another chemical that we're interested in. We use tools from synthetic biology and systems biology in this process here. So why yeast? Well, as I said, it's used extensively, but it's probably also the organism that we know the most of. This is hard to quantify, and of course I may be biased, but we have an immense amount of information because it has been used not only for industrial application for many years, but yeast is also used as a model organism for studying many fundamental processes. So Jack Szostak, who was talking yesterday, he actually got the Nobel Prize for studying telomeres in yeast. And there are many Nobel laureates who studied yeast, used yeast as a model organism. So that's why we have all this knowledge here. It's very robust industrially. It's already applied. It's genetically tractable. And it also has this characteristic of what we call generally regarded as safe. And that comes because it's been used for so many years to make food products. It's already used for many different products, including insulin and vaccines, and is ongoing for development of many other products here. So let me give a couple of examples of what we have done with engineering yeast. So one thing is to produce perfumes. So what we do is that we take genetic materials from plants, we put it into yeast, as well as we engineer parts of yeast endogenous metabolism, so that we say that we deregulate enzymes, we overexpress certain enzymes, so that we efficiently can convert the sugar, the glucose, into the product of interest, for example, xanthine, which is in sandalwood, used in many perfumes. And then we can extract these from the medium and we can get them. So why would you do that when you can just extract them from plants? Well, in many cases, these plants don't grow very well, except for maybe very rare places. And so now you can produce these chemicals in a scalable fashion, and you can also get them in amounts that is basically, suiting your need. And so we did this in collaboration with a Swiss company, Firmenich, which is one of the largest companies in the world to make these kind of ingredients. And they have actually now taken this technology and is commercially producing several of these chemicals. And so when you buy perfumes in the future, they may be produced by yeast. Resveratrol is another example. This is an antioxidant that is found in the skin of grapes, and it's associated with a number of health properties, health claims. It's also used in cosmetics. Many of you will have seen that you can have creams that are anti-aging, often because there's added resveratrol in it. It's kind of tough to extract it from grapes, so it's difficult to get enough of it. So we engineered yeast. Again, we took genes from plants as well as from other sources, and then reconstructed the pathway to make resveratrol, and we could now produce significant amount of it. And this process is actually today also commercialized by a company, Evolva. They actually took over a company that I founded initially that developed the process for this. Fatty acid production. There's a lot of interest in producing fatty acids because they're very similar in structure to hydrocarbons. So they can be used as fuels, lubricants, and cosmetics. And the objective would, of course, be eventually to be able to fly on biofuels. What we did was that and I'm not going to go into the details of this, but we did multiple changes and completely rewrote the metabolism, fatty acid metabolism in yeast in order to efficiently produce these. And so we can now produce more than 10 grams per liter of these fatty acids, and that is a good starting point for actually converting these further on to make various different chemicals. So that's fine. These were the fatty acids that yeast normally makes. These has a chain length of 16 carbon atoms or 18 carbon atoms. But the chemical industry is also interested to have short-chain fatty acids. So they are maybe C8, C10, or C12 carbon atoms. And so what we did was that we looked into the enzyme that is producing fatty acids. This is a fascinating enzyme. It's a multifunctional enzyme. It's huge, and it do a number of different catalytic steps in one unit. So it's like a whole little pathway by itself. But we identified that we could actually insert another enzyme inside the catalytic site of this multifunctional enzyme. And so that when the carbon chain has reached 8 or 10, there is an enzyme that cleaves it off and it leaves the enzyme. It's a so-called thioesterase. And hereby we could produce C8 and C10 fatty acids. And again, these are much more attractive to be used in gasoline. The energy content is much higher than ethanol, as you can see. And this technology we licensed to Total, a French energy giant. But we're also looking into using these short-chain fatty acids for different chemical use in our own startup, Biopetroleum. We can also go the other way and produce longer fatty acids. They are hard to get because they are only available in a few plants. One example is, for example, in jojoba plants. They have these very long-chain fatty acids. So we extended, again, an engineered yeast metabolism. We also took an enzyme from a bacteria and inserted, so we can now produce these very, very long-chain fatty acids, as you can see, and also their associated alcohols here. And as I said, one application of these is in cosmetics, for example, in jojoba oils. Some of you may be familiar with that, but it can also be used as lubricants. We've also been working on making yeast more efficient to make biopharmaceuticals. We have taken insulin as an example. And what we did was here we take a so-called proinsulin. Insulin has actually 2 chains, but here we have a peptide bond, a peptide connecting the 2 peptide chains. And what we did was that we used microfluidics to screen. And so here we were saying we need to learn something new. So we created a mutagenesis library. We then used microfluidic screening to identify yeast strain that was secreting more insulin. And then we sequenced these strains, and then we mapped out mutations, how they are associated with. And this gives us a lot of new insight into how this relatively complex system actually functions, namely the secretion pathway in yeast. So let me just end with not so much about cell factories, but just a study that we recently did in order to address one of these problems I referred to about antibiotic resistance. So what we did there in order to find new potentially chemicals was that we sequenced 26 fungal strains. They're all from this class here, a family of Penicillia. So not surprisingly, they are also, one of them is also producing penicillin. What was fascinating that we found was that when we look into the different families of enzymes that are present in these species here is that the most abundant family is associated with making what we call secondary metabolites, complex chemicals that have potential for different application as bioactives. And actually, when we look into, on average, on these so-called biosynthetic gene clusters, so these are the set of genes that are producing these secondary metabolites, there are close to 60 of these present in each species. That's an immense capacity, actually. So why have we not found this when we're kind of screening these different fungi? Well, first of all, many people have not looked into these fungi before, but also many of these biosynthetic gene clusters are so-called silent. That means that they are not expressed. But now we have them, and we can begin to express them in yeast and explore the capability of producing new chemicals. Here's just an illustration about how you can also see these gene clusters, that they are really having multiple functions, several different enzymes that are doing variations, and we can begin to actually use genomics to get insight into the function of these. So I'm really confident that we in the future will see that we will use various energy sources. We will also have biorefineries that will provide liquid transportation fuels as well as chemicals, and we'll rely on Francis's efficient enzymes in this process also, so we can get all the materials and energy sources that we are needing for having a more sustainable society in the future. Just one last advocate for the use of biofuels, because there's a lot of discussion about this. And there was a recent paper in Nature where they did blending of biofuels, 50% actually, into airplanes, and then they measured the particle emission from the airplane that was flying on respectively biofuel blended or regularly jet fuel. And they found a significant reduction in particle emission that is very much associated also with greenhouse gas development. global warming. And you can even see it here on the picture here. This is the biofuel plane here. This is the non-biofuel plane. You really have significant reduction. So can we use our knowledge on yeast metabolism to study human diseases? So you heard a great talk from Lee Hood this morning about personalized medicine. Really the objective is to go from this basic scenario here with disease group to control group to actually stratify subjects into individual groups that are better clarified and how we can treat them better so we don't end up in this situation. We're giving a lot of drugs to people that they actually don't need and it doesn't cure their disease. But for this stratification here, we need new biomarkers. And let me focus particularly on this. And so for this, we're using these models that I referred to that are capturing information about the metabolism in all the different cells. So we are writing down the essence of the reaction. So we have all the individual stoichiometries. We're linking that to enzymes. That gives us this metabolic network. And I showed you earlier on the metabolic network for yeast, how it looks globally. You can imagine for human cells with many more reactions and many more genes and enzymes, it's even more complex. But now we have a scaffold, and we can use this scaffold for analysis. And so just some numbers for the human model. We have more than 8,000 reactions here. You can see, and actually close to 4,000 genes associated with this. And we can have models that are specific for different tissues. We call the general model HMR2, human metabolic reaction. But we can also look into what is the reactions present, for example, in liver or muscle or different types of tissues. And so we did this globally. This was actually done by Adil Maldinoglu, who's also here in the auditorium. And he mapped, he looked at, so let's take this generic model and then see what reactions, which enzymes are expressed in different tissues, and in this case, 32 different tissues across here. And red means that this reaction was present in that tissue. And then you can see there's one tissue that is sticking out here. It almost has everything. That's liver. Our liver is really good in metabolizing stuff. It actually has many of these P450 enzymes that Francis was also talking about that are used to degrade different toxic. But then you can see there are some variations. Some tissues have specified metabolism here. These are kind of parts of metabolic pathways that are active in these tissues and so on. So we can begin to see what is actually going on in different tissues. We can also use and say, now we take transcription data from a tumor versus a healthy control, and then see what part of metabolism is particularly changing. That could lead to a certain parts of metabolism that is maybe upregulated in the tumor and downregulated in the tumor. And we could then begin to look for some of the chemicals, the metabolites that are associated with that pathway, and they could maybe be biomarkers. Maybe we would see then that those metabolic pathways that are upregulated in the tumor, also the metabolites associated with that pathway are found at higher levels in blood, for example, of those subjects. So in order to evaluate whether we could test this or see this, we did an analysis and started with looking into the expression in 10 tumors, and actually we set out because we wanted to see if there was any big differences between the metabolism of these tumors. But we got sidetracked because we found that one type of tumor, one type of cancer was sticking out really distinctly from the other cancers, the other 9 cancers, and this was what is called clear cell renal carcinoma. It's the most typical form of kidney cancer. So we looked further into that, and it turns out that in this tumor, in this cancer, that is associated with a very distinct deregulation of one particular pathway. And that pathway is involved in making what we call these glycosaminoglycans. This is a set of metabolites that are normally used to degrade proteins, but we find them really changed in the tumor here. And we can see here red means that the corresponding gene was upregulated, blue means that it was down, that it's kind of a complex, and all the connections is because these enzymes are using different substrates and highly connected. But it becomes a little bit more clear when we lay it out in a traditional pathway map, because suddenly we can see, wow, this pathway here leading to this set of chemicals here is red, that means up, so that's upregulated. This one here is down. So we could maybe expect that some of these chemicals over here were higher level in urine and blood, and these ones were lower. We validated some of these with proteomic staining, which you would have to do in order to prove that this is not just the transcript, but also carried over to the protein. And so we did that. We measured glycosaminoglycans in plasma and urine in patients that had clear cell renal carcinoma versus healthy controls. And here's just an illustration of some of these measurements across several different types of measurements here. But what is interesting is that when we take all these measurements and then develop a computer code, a computer score for marking this. So this is a computer generated saying that maybe we take this metabolite and we multiply it by 5 plus this one multiplied by maybe 7 divided by this one divided by 12 or multiplied by 12 or something like that. I mean, the computer generates automatically. And then we get a score based on these measurements. And here you can see this is for the patients that had a metastatic clear cell renal carcinoma. That means the cancer has gone crazy and wild. It's spreading to the human body. And here you have healthy. And this is here if you combine measurement in both plasma and urine. But both plasma and urine also, you can see the measurements, the score is much, much higher in the patients here. We can also develop this so-called ROC curve that is a predictor of how good this marker is for actually identification. And we validated this in a separate cohort. So how could you use this? Well, for example, when you are getting treated, maybe you're not responding to this drug. We can now see this because the tumor is still active. So, and actually more than 60% of the patients don't respond to the first line of drug. Also, when you come into the clinic and you have a renal mass, actually in 20% of the cases, people are being operated even though the tumor is not active. So those operations could be spared. At least we could put those people under control and monitor to see if the tumor would progress and become non-benign. We can also use it for testing people that have been operated and follow them up and see if actually the tumor, everything has been taken away or whether it develops into so-called metastatic cancer. It can also, of course, be used for early diagnosis and actually for general screening. There are some challenges with this that we can also discuss. So it turns out that this is, even though we were led down that track by this cancer being special metabolically, but it turns out that this reprogramming of glycosaminoglycan metabolism is not unique to kidney cancer. It's also in another cancer here. Here's shown again the plasma, urine, and the combined score. And again, you can completely separate. And it turns out that this score is different from the one, even though the measurements are the same, but the score is different from that from kidney cancer. So we can measure these metabolites in plasma and urine, and we can actually say which type of cancer that is likely to be present in that subject. This is shown here. When we take the score for this cancer and use it for the data for kidney, you can see it's not sticking out, whereas if we do the other way around, this is sticking out here. So let me end to talk a little bit about the human gut microbiome. This is a fascinating thing. We have about 2 kilos of bacteria in our human gut, and they are doing all kind of crosstalk with us. I mean, I don't think we even want to know all the things that they're doing to us, but we are kind of dependent on them, and we have about 10 times more bacteria than we have human cells in the body. They are changing over time. There's much interested in that also to follow how that changes are going, and that's actually also part of Lee's wellness program to follow that change. And honestly, we don't really know what they are doing, but we find a lot of links to diseases. Here's a list of diseases: obesity, malnutrition, allergies, diabetes, bowel disease, colon cancer, heart disease. The list is long, even autism and depression. And we don't know exactly what is the cause, but this is what we can begin to study and go into. So as I said, most of these studies are correlative studies. We don't have any mechanisms, and so we really need causal information to get to this. And so if we get causal information, we can of course also begin to get an opportunity to intervene. We can begin to give bugs to people to cure them. So how do we study this? We do this by sequencing. We take the microbial community, we extract total DNA, not from one bug, but now we have thousands of bugs there. And then we annotate this, and as we are so much in love with metabolism, we look at particularly into what metabolism does these bacteria do. This is really big data. We get a lot of data typically, so you can see here 3 to the power 10 to the 11th base pairs that we have to file through. Here's just an example of this correlation stuff. Here is then all the different bacteria that we identified in subjects, and we were looking into subjects that have either type 2 diabetes or healthy controls. And then we were correlating what is the abundance of, for example, this bacteria here. If you have high level of this bacteria, you normally also have high level of many of these plasma markers here. Some of these here are bad, high insulin levels, for example, bad fasting glucose reference, and so on. So this is what we call a bad profile. If you have high levels of these, you're likely to have elevated levels of these. These ones here are kind of a good profile, and these are present more in healthy subjects. So you can imagine we could begin to pick out, but still the list is long, right? But we could begin to pick out some of these guys here and put into those that have diabetes and hope that they can get cured. Well, you can't do that because you need still to have causal relationship. So how do we do that? This is a quite complicated study we did here, but we actually used models of the different species in the gut, and then we looked at how are people responding differently to diet changes. And so we had 2 groups of subjects, one that was responding very well and one that was not responding so much to a dietary restriction. And what we could model, correctly predict, was that the amino acid levels in the blood was really associated with these changes here. And if you don't have the right bacterial composition, you don't respond very well to dietary changes because they will impact this. And actually many of these amino acids here, levels of these are also associated with disease development, as we also could find. So people talk about these thousands of different bacteria. So we did an analysis recently where we were looking into, are there really that many? Yes, there is. There is really a lot. But it maybe doesn't matter so much because most of them are present in very low amounts. And so we therefore now are focusing in on what we call generalist and specialist. There's a set of core that is present in most of us. Maybe there's some small variations, but they seem to be consistent. And you can see they are making up, some of them are really getting close to 10% of the abundance. Maybe not exactly, but you know, you can see maybe around 1%, and there are a set of cores that are about 100 species. And then we have some generalists and some specialists, and then we have all the rest. And we're beginning to focus in and zoom in on these guys here. And we are then looking into the core occurrence between these. So which ones are, for example, connected and always present in all the subjects that are normal glucose tolerant versus those that have type 2 diabetes. And these networks can begin to tell us something about why these species live together, and now we can study what metabolism they have. So let me end with this slide here, coming back to say, can we begin to use this information to solve a fundamental problem in Africa and other places with malnutrition? And the problem with many malnourished children is that they are not even responding when they are given good food. This what is often referred to as ready-to-use therapeutic foods. So they don't gain weight, they don't really grow, even if they're giving this. And so what we did was that we looked into the gut bacterial composition in children, and we compared that with the Swedish cohort of children. And what we found was that the Malawian, and also in this case Bangladeshi children that are responding poorly to these therapeutic foods, they actually miss the capability of producing many essential amino acids. And so they simply don't turn over the food correctly, and the children don't get the essential amino acids, which is important for their muscles and proteins in the body generally to grow. And we're looking into this with the Bill and Melinda Gates Foundation. And so what we would have to do possibly in the future is to combine these ready-to-use therapeutic foods together with provision of specific bacteria that actually can do this. So understanding metabolism allows you to address many of the grand challenges of our future, so enjoy it. Thank the people here, yeah. Thank you.